Okay. Hello, we're the fMRIusic team, part of the Jahanika group. Our project focuses on preprocessing fMRI data for analyzing emotional responses. I am Aryan.
And I'm Monami.
Functional magnetic resonance imaging, or fMRI, is a scan that measures brain activity by detecting changes associated with blood flow. This technique relies on the fact that cerebral blood flow and neuronal activation are coupled together. For example, when an area of the brain is in use, blood flow to that region increases as it is used more and more. It uses MR imaging to measure tiny changes in blood flow. It also measures blood oxygen levels. fMRI examines the brain's functional anatomy, evaluates the effects of stroke or other diseases, and detects abnormalities within the brain that cannot be found with other techniques. The brain's auditory network works via auditory messages that are conveyed to the brain. The auditory network is across both hemispheres of the brain and is located on the superior portion of the temporal lobe of each hemisphere, and it is also interconnected. The primary auditory pathway exclusively carries messages, while the non-primary pathway carries all types of sensory messages. If your auditory cortex is damaged, then it can lead to partial or complete deafness. The primary way we can view fMRI data is through a comprehensive neuroimaging analysis library called FSL, which includes various software programs that help in analyzing fMRI, MRI, and diffusion MRI data. It contains ICA-Melodic, which is a software that runs individual component analysis as part of our preprocessing procedure. However, in terms of viewing fMRI data, the FSL primarily supplies us with FSLI. The pictures on the right are functional and anatomical images of the subject's brain. The anatomical image shows us the structure of the subject's brain, and as you can see in the image on the top right, there is a black and white image of the brain, and the lighter regions signify the white matter, and the darker regions signify the gray matter of the brain. The white matter are the myelinated regions of the brain, while the gray matter are the regions that are not. The presence of the white and gray matter indicate many factors regarding the brain, such as specific regions, age, or even its current health conditions. What our study is arguably more concerned with are the functional images of the patient's brain. As you can see on the bottom right, the functional images have generally lower resolutions. However, it should still be noted that the fMRI is still one of the best neuroimaging tools in terms of spatial resolution, although it falls behind on temporal resolution compared to tools like the positron emission topography. fMRI tracks the level of oxygen in different parts of the brain, which is shown on the functional image through color and through multiple processing steps. We can also view clear images that show the brain regions that are active.
What happened?
Before we talk about the pre-processing steps our team went through, we should talk about the specific format our data set was in.
Aryan, I think we can wait. Kirtana, can you mute yourself, please?
I'm sorry, honey. She might not like them because they're broken, but no,
You're still not muted, Kirtana.
You can't give them to mom. Dad had me type.
Kirtana, can you mute yourself, please?
I feel like the dad's not like a good emotion.
I think the person that started the meeting could mute any participant.
I mean, David is not. Oh, I'll try to text him.
The dad. Stop. Really?
Whenever he leaves, he should give host control over to whoever's leaving.
That's so. Yeah. I know what kind of dad says? Mom.
Kirtana, are you here? Can you mute yourself, Kirtana?
Hey, Sahar, let me assign the host to you. So if anyone does that, you can do that.
Thank you so much. Thank you, David.
Sorry about that. It does happen that way. All right, so you're officially the host.
Thank
So you
you.
can start recording, stop, do whatever you need to.
Thank you, sir. Thank you.
You're welcome. I mute people all the time.
Go ahead, Aryan.
Okay. Before we talk about the pre-processing steps our team went through, we should also talk about the specific format our dataset was in and how the files were organized. First and foremost, the pure fMRI data files that we used were in NIfTY format. There are two primarily data types that are commonly used in the field of neuroimaging, and they're DICOM and NIfTY. DICOM, which stands for Digital Imaging and Communications in Medicine, are essentially 2D images of medical scans. NIfTY files are neuroimaging informatics technology initiatives, and they are 3D data types and are accompanied by JSON files and BIDs. In total, our NIfTY files were organized according to the BID system, and that is a standardized file organization format. In this format, files are organized into different tasks and different neuroimaging techniques, and ultimately different patients or different subjects. Next one. Our dataset is a structural dataset from the paper called Dynamic Intersubject Neural Synchronization, Reflects Affective Responses to Sad Music. And this data explores the brain activity in 39 subjects listening to music. The brain activity was analyzed from playing happy and sad clips of music with or without audio. We took the data and converted the functional data and uploaded to the ASDRP server. The functional data measures the neuron activity, while the structural data measures the anatomical structure.
All right, so in our attempts to pre-process, the main tool we used was fMRIprep. fMRIprep is an fMRI data pre-processing pipeline developed by the neuroimaging pre-processing tools, or NIPREPs, and uses a combination of multiple pre-processing tools such as FreeSurfer, AFNI, FSL, and ANTS to perform structural and functional pre-processing on raw fMRI data. This pre-processing step allows us to remove noise and fix issues due to motion, such as head movement. fMRIprep, along with many neuroimaging pre-processing pipelines, requires several dependencies, as mentioned above, with different libraries which might have different compatibility issues with the operating system and such. The solution to this was creating an isolated environment of an OS-level visualization called a container through the Docker tool. As you can see here, fMRIprep offers several pre-processing steps that help to clarify and make out significant fMRI changes or differences in fMRI data, and to essentially just make it more clear in both the structural and functional sense. Our other softwares that we used included MicroGL, which converts DICOM to NIfTY files, as Aryan mentioned before, and which basically helps us to adhere to the BIDS format necessary for fMRIprep. FSL is also a very useful neuroimaging library, and from FSL, we used Melodic and FSL-Eyes, which are programs to run individual component analysis and to view fMRI data. So these are some of the examples of fMRIprep outputs. So in the upper left corner, you can see a heat map summarizing the correlation structure among confound variables. And what confound variables are is just they're variables that explain activity that isn't necessarily related to the tasks that the subjects were undergoing when the scan was occurring. And to the right of that heat map is a graph showing the magnitude of these correlations. On the bottom is an anatomical preprocessing step called spatial normalization. Here, the patient's brain is essentially mapped onto a reference or template of previously collected brain scans that were compiled into an average brain template, which is the one we used was MNI 152. So to summarize what our results ended up being and what we've done so far using fMRIprep, we've successfully implemented fMRIprep on 39 subjects, and this produced field map correction, head motion correction, and susceptibility distortion estimation derivatives, which help us to correct distortion between functional and anatomical data. These were all outputted into a derivatives directory on our server, and they essentially allow us to pre-process and remove unwanted artifacts that could affect our results in the future. So, along with fMRIprep, it produces a HTML file that really helps us to be able to determine the success of the command we ran. This HTML file allows us to go through every subject's fMRIprep outputs and evaluate whether it is to move on to the ready to move on to the next step for us, which is ICA-Aroma. The HTML file contains outputs of different pre-processing functions, such as head motion correction, susceptibility distortion correction, in addition to visualization of data. We consulted various external sources in order to comply on a set of guidelines that we'd use to determine if our data was appropriate to be and ready to move on to the next step. Some of these guidelines included if the segmentation was done correctly. So if the identification of gray versus white matter was done properly. So red outlines should typically surround the entire brain, and the blue outlines should separate the gray and white matter. And of course, if the susceptibility distortion correction should fit these red outlines, as you'll see later on in the next few slides. And so a major problem we encountered with several subjects was with susceptibility distortion correction, which essentially corrects the functional image to fit the anatomical outline. We found that the correction morphed the brain unnaturally in several patients, sometimes producing unnatural protrusions or just completely ignoring the border of the brain. In this picture, you'll see the anatomical segmentation. And this step was typically very accurate, or pretty accurate from what we could see when we ran fMRIprep. And the functional data tended to have a lot more issues, which you'll see in these slides. Here you can see in the distorted and corrected, the distorted is on the top and the corrected is on the bottom. You can see that even though the distorted and corrected data still seem to have a lot of protrusions in them, as you can see on the lower left picture. And then on the lower right, you can see, although it's very faint, that the red line doesn't completely encompass the brain, and that can really hinder our results. Here's a more successful fMRIprep preprocessing in which the brain was more clearly identified and the anatomical and functional brain mapping was more accurate. There are still some issues with this. However, we've deemed that if after quality control, the fMRIprep outputs are satisfactory, we just move on to the next step. Otherwise, we'll rerun the subjects again in hopes that the command will be able to reconfigure and reestablish a better outline and fix the issues we saw earlier. And the next step, as I mentioned, is ICA-Aroma, more specifically, melodic step of this process.
ICA-Aroma is a data-driven method to identify and remove motion-related independent components from fMRI data. It exploits a small, robust set of features which provides direct and easy applicability. ICA-Aroma removes artifacts, separates sources of the brain signals, and extracts noise. ICA Melodic uses independent component analysis, or ICA, to decompose a single or multiple 4D datasets into different spatial and temporal components. For ICA group analysis, Melodic either uses tensorial independent component analysis, or TICA, where data is decomposed into spatial maps, time courses, and subject/session models, or a simpler temporal connotation approach. Melodic can pick out different activation and artifactual components without any explicit time series model being specified from a tool from FSL. We aimed at selecting it from the melodic output. C-PAC, or the configurable pipeline for analysis of connectomes, is a software pipeline for automated preprocessing and analysis of resting state fMRI data. It builds upon existing software packages, including AFNI, FSL, and AMP. C-PAC makes it easier for users and experts to explore data using a wide array of analytic tools. C-PAC is configurable, open source, and NiPi and Python based. The main goal of C-PAC is to build off the NiPi platform to form an automated processing pipeline for resting state MRI data. C-PAC is optimized for large datasets and is able to analyze hundreds of subjects. Our main goal for C-PAC is to complete time series analysis, which will extract a signal measured at each voxel across the entire run, which helps determine the strength of the neural activity. So far, we have successfully implemented fMRIprep on Docker, and we have also completed preprocessing on all 39 subjects. We have quality controlled most of these subjects, and the rest are being done as we speak. As we have also figured out a way to run melodic ICA for the subjects for the final step of preprocessing. Currently, we are testing the C-PAC pipeline for the time series extraction.
Upon finishing up our last few pre-processing steps and tying all loose ends, we'll figure out our C-PAC pipeline and be able to successfully extract our time series data. With this time series data, we'll be able to run second level analysis, which is a more cohesive group level analysis with machine learning algorithms for data analysis. And through these results, we hope to be able to determine stimulus-driven impacts and changes to activity and connectivity. For our acknowledgements, we would like to thank our PI, Saharjan Ikia, for all the support and guidance she has given us through this project, as it would not have been possible without her. We'd also like to thank ASDRP for providing us with the resources to perform this research. And finally, we'd like to thank our fellow researchers in our team, Jongmin An, Ayan Khan, Shreya Arvind, and of course, our past members who've been a great help and will continue to be a great help in this project. Thank you for listening to our presentation, and the floor is open for questions.
Thank you so much. Just to emphasize, trust me, this is a project that a postdoc candidates at High Neuroimaging Center will work on it for their thesis. PhD students, they'll work on it, postdoc will work on it. This is like the pre-processing pipeline to pre-process neuroimaging data on and run it on Docker is really challenging. So I want that we all give credit to this hardworking team, the old members, new members. There's some old members that they have left, but the current members. So this is definitely a challenging project. I want to give them a lot of credit for not giving up and trying hard. And wish them best of luck. Thank you, Morami. Thank you, Aryan. So they are open to questions.
Ashwin?
So you can come out of the mute and ask them.
Okay, cool. So my question was, what was the significance of the differences between the white and gray matter? Like I may have missed something, but I wasn't exactly sure like why that mattered.
Oh, sure. So I guess the best, or does Aryan want to take this question? Here, hold on. I think maybe I should reshare the slides. Sorry, one sec. Let me reshare the slides. While I try to figure out how to reshare the slides, basically the white matter is myelinated, and that just means that white matter is more, it's basically the important, I don't want to say the important part of the brain, but the neurons that deal with actual activity and like transfer of basically neural activity in the brain. So white matter is what controls, quote unquote, like our breathing, our heart rates, things like that. So gray matter is more related to protecting the white matter, and also for basically cleaning up the brain, if in like simple terms, if I want to put it that way. Yeah. So basically the activity we're looking for would most likely be in the white matter, and we want to be able to differentiate between the two because obviously the cells in the white matter are undergoing metabolism, and that has changes in blood oxygen levels, and that would be detected by the MRI, but we want to be able to say that, oh, this is not related to our activity, it's just the brain itself doing what it needs to do to be able to run and function on its own. Yeah.
Got it. Okay, so it was like, I suppose, establishing a control and knowing where to look. Okay, cool. Thank you.
Do we have any other question? So if you are interested on neuroimaging topics and techniques, next 2 weeks we'll be having 2 EEG teams from my lab presenting their work. They started their project during summer term, during the fall term. So they are pretty, the project is pretty, I would say, new, but they have done amazing progress. So if you do not have any question, I think we can wrap up our colloquia tonight by 7:30. No? Okay. Wish everyone have a good night. Thank you, Aryan and Monami again. And best of luck. Thank you, everyone.